Bacterial genome sequencing identification of the source of outbreaks. In my book, Bacterial Genetics and Genomics, in Chapter 11, I discuss how we use DNA within bacteria to identify bacterial outbreaks and understand how those outbreaks occurred, how they're spread and transmitted, where they've come from, and maybe how we can control them. So in my book, where I discuss one of the outbreaks that has been understood using genome sequencing, so understanding the DNA within the bacteria. In January 2010, there was an earthquake that devastated Haiti. The international community sent aid workers and supplies to Haiti to be able to help the people of Haiti. In the aftermath of that earthquake, however, Haiti had its first cholera outbreak in nearly 100 years. So Haiti hadn't seen cholera for a very long time, and then all of a sudden, after the earthquake in October, then cholera started appearing across the nation of Haiti. So this is what cholera aid would look like when the WHO recommends how to take care of people who have cholera. So there are treatment kits that are brought in, there's laboratory investigation kits that are brought in, and hardware kits that are brought in to help when there is a cholera outbreak. There are an estimated 4 million cases and 143,000 deaths from cholera worldwide each year, and these estimates are from 2017. So cholera is a big problem worldwide, and when we have these outbreaks, then the questions are, what has happened that has brought the cholera into this region, and how has this occurred? So one of the ways that that can be investigated is using genome sequencing of the bacteria. And those bacterial cells are shown there. There's these little squiggly kind of rod-shaped cells. In October 2010, 10 months after the earthquake occurred, patients were found in 2 different regions of Haiti with the bacteria Vibrio cholera. Within a few weeks, cases had spread across Haiti, including cases in the capital, Port-au-Prince. The earthquake killed more than 200,000 people. And within a few weeks, cholera killed 900 people and hospitalized over 16,000. Within 2 years, it was estimated that more than 7,400 people had died from cholera in Haiti, with 330,000 people having been hospitalized and over 600,000 people having been infected. So within just 2 years of when the cholera started in Haiti. There was a UN stabilization mission in Haiti at the time of the earthquake. And so there's an abbreviation there for the stabilization mission. MINUSTA was established in 2004 after the removal of the Haitian president. These MINUSTA troops provided aid immediately after the earthquake. They helped to clear the debris, rebuild the infrastructure of Haiti, and maintain law and security in the aftermath of the earthquake. Troops included troops worldwide who came to the aid of the Haitians after this devastating earthquake. And different troops were brought in to relieve troops that had been there for quite some time. So troops came from Nepal, arriving in Haiti in October 2010 to take over their shift of helping out the people in Haiti. Unfortunately, the first cases of cholera also appeared in October 2010. Cholera is endemic in Nepal. The MINUSTA troops had no symptoms, however, when they went to Haiti. So no one suspected that these troops that were arriving to help in Haiti might be carrying cholera. However, cholera can be carried asymptomatically, and it looks like the Nepalese MINUSTA troops probably carried the cholera with them, just looking at it from a microbiological sense. The Nepalese MINUSTA troops were not tested for Vibrio cholera, but nor were any other troops. So then the question was, could it be these troops that had brought the cholera into Haiti? So this is where we bring in the genetic testing. The Haiti strains of Vibrio cholera were genome sequenced and they were typed, and it was discovered that the Vibrio cholera in Haiti was V. cholera 01, and this is not a type that is found in the Western Hemisphere. V. cholera 01 is found in Southeast Asia. Further testing was done, and the genome sequence data to understand the individual characteristics of the DNA within the Haitian cholera showed that the DNA from these bacteria is nearly identical to cholera bacteria from Nepal in the summer of 2010. Using that genome sequencing, then there was a definitive link that identified the aid workers that had come from Nepal with the Haitian outbreak of cholera. The outbreak originated in Haiti in Maye, where Nepalese MINUSTA troops were stationed. The MINUSTA base was near the Maye tributary, which feeds the Artibonite River. There were problems at the base with waste containment, which caused fecal contamination into Haiti's largest river, the source of water for drinking, for bathing, and cooking. Therefore, when the troops asymptomatically carrying cholera arrived, the conditions were in place for it to spread. So this natural disaster and subsequent outbreaks emphasized that there's a prioritization of sanitation during disaster planning and response that needs to happen, and an understanding that the post-disaster epidemics may not be endemic. So the diseases that we see occur, those infectious diseases that may happen after a disaster, may not be infections that are endemic to the region. They may be things that are introduced by people who have come to help. So vaccination, chemoprophylaxis, and screening of disaster responders is quite important. The last case of cholera in Haiti was in January 2019, so it has taken quite some time to get that cholera outbreak under control in Haiti. If you'd like to learn more about bacterial genetics and genomics, you can check out my book, available from CRC Press, Taylor and Francis.